for semester 3. Well, at the outset, congratulations to all of you. You all have gone in M.Sc Part 2 program. And that's how we are using this time available to us for scheduling your lectures, so that theory topics, if we deal with at ease, with time and comfortably in this period, later on when we meet face to face, we can always have more scholarly interaction, and we can also devote much time for the practicals. Irrespective of your specialization, you have been asked to attend all lectures for the simple reason that no matter what your specialization is, the model system that you are investigating in your undergraduate and postgraduate study is a plant system. And therefore, knowing a model system in and out with reference to all the peculiarities is always very helpful. And that's the reason we all are attending all lectures. Today, I start the lectures with the topic on stress physiology, part of the syllabus for physiology students as paper 4, unit 2 and unit 1, both are dedicated to stress physiology. But as I said, it's a learning process for all of us. What is stress? Any deviation from normal condition can be referred to as stress. With reference to plants, plant growth and development, we'll put it like this: any deviation from normal growth and development of the plant can be a factor considered as stress. I'll give you one example and explain this. Green plants are gifted with an ability to photosynthesize and synthesize glucose and other metabolites, not only for its own growth and development, but also for the other trophic levels of the ecosystem, because green plants are the primary producers at the base of the ecological pyramid. Now, this photosynthetic ability of green plants is controlled by a variety of factors: light, temperature, humidity, CO2 concentration, O2 concentration, stomatal size, size of the leaf, number of leaves, orientation of leaves, thickness of the leaves, palisade to spongy ratio in the leaves, number of chloroplast, structure of chloroplast, distribution of chloroplast, pigment system composition, ability of a plant to avoid photooxidation, etc., etc. Now, of all these factors, if I have to handpick 4 environmental factors which affects the plant photosynthesis, then I would choose light, temperature, humidity, and carbon dioxide concentration. If any of these factors are available in amounts less than what is required for the normal optimum photosynthesis of the plant, then we will say that the plant is suffering from stress. These factors being environmental factor, we will say plant is suffering from environmental stress. Climatic factors, so climatic stress. External factors, so external stress. Physical factors, so physical stress. Well, will plants respond to this stress? Yes. Every living organism, as per its capacity, responds to the stress. How will plant respond to this stress? Says the factor that we are discussing is controlling the photosynthesis of the plant. Under adverse condition of this factor, therefore, the plant photosynthesis will drop down. Now, decrease in photosynthesis means decrease in glucose production, and glucose being the central metabolite, vital metabolite, all primary metabolic pathways will drop down. Primary metabolic pathways supply precursors for secondary metabolic pathways, and secondary metabolic pathways, when branch out, they give you important enzyme cofactors, vitamins, etc. So ultimately, entire metabolite makeup of the plant contributing to the productivity of the plant will suffer. So can I summarize whatever I said so far as external physical stress gets translated into internal physiological stress, and as an outcome of this, plant growth and development suffers. Now, variety of factors that controls the plant growth can be broadly divided into two kind of factors: biotic factor and abiotic factor. And if they happen to be the stress factor, then I say I'm talking about two kind of stresses: biotic stress and abiotic stress. Biotic stress is the stress which is caused in plants by other living organisms like bacteria, viruses, fungi, nematodes, insects. Because when these organisms infect the plant will have to give a very quick response to this infection. Why? Because the causing agent is directly depriving the host plant of its own nutrients. See, these pathogens survive on the nutrients which a plant is producing. Something that the plant was producing for itself is now being totally taken up by these infecting organisms. So the plant will slowly be depriving its own self with those. And if it doesn't give a very quick response, then it may lead to the death of the plant. And that is why any response to a biotic stress is a quick response. Moreover, the main target site of any biotic stress is the leaves, because there itself is the plenty of food which is not only synthesized but available for them. In contrast to this, when you talk about the second type of stress called abiotic stress, it is the stress that is caused by non-biological agents, the environmental and the adaptive factors. Say, for example, temperature, light, water, soil. Too many extremes of it are harmful. For example, too high a temperature will lead to desiccation and will lead to drought. Same way, too low a temperature will also be harmful to the plant because it will develop what is known as a freezing stress. The cytoplasmic water will freeze and therefore it will not be available for the metabolic activities. Too less of a water will again cause the drying condition, osmotic stress, and the plant will be subjected to drought. Or too high of a water can also lead to flooding stress. Inside the soil, no nutrients can cause nutritional stress to the plant, and having high amount of salts can cause salinity, and thereby a plant is subjected to salinity stress. Now, one point that I would like to bring to your notice is that when a abiotic stress is experienced by the plant, the target site is root. Because if you understand, everything in the plant is rooted through roots. But in the biotic stress, we saw leaf is the main site. Second, biotic stress manifests, unfolds very fast, and therefore the plant response to it should also be very fast. Compared to this, abiotic stress is a stress which is of the prolonged type. In comparison, don't say that biotic is fast and abiotic is slow. It is not slow. It is spreaded over a longer period of time, so it becomes prolonged. But it is in a way advantageous to the plant that plant can have mechanisms developed temporarily or permanently to mitigate, to manage, to adjust, to adapt itself to this stress. Now, please bear one thing in mind. Unlike other living creatures like humans and animals, plants are not freely moving inside the soil. They are stationary. And therefore, the way humans or animals can migrate away from the non-conducive stress condition to comfortable non-stress condition, plants cannot do that. They are always left with only two choices: either succumb to the stress and die, or adapt yourself to the stress. Now, in order to know, therefore, that the threat is on its way, or I am being approached by a threat of stress, plant has to have a very defined mechanism so that it can prepare itself for the coming threat. What I am trying to draw your attention to is the point that plants have a very well defined mechanism right from identifying the stimulus, responding to stimulus, and developing method to take care of the stimulus. The stimulus which is actually received, a stimulus of stress, which is actually received, is first sensed by sensors, which are located on the cell surface or which could be located in the cytoplasm. Once these sensors sense the stimulus, this stimulus is then transferred to transcriptionary machinery, which is situated inside the nucleus. And thus, with the help of different signal transduction pathways, differential transcriptional responses are generated by the plant in response to the stress. I repeat, signaling of the stress is identified by sensors in the cytoplasm, which then directly transfer the stimulus to the transcriptional factors available in nucleus, which starts generating the response in terms of differential transcriptional proteins via means of signal transduction pathways. So it means that signal transduction pathways act as a link between the stress environment and the response, appropriate biochemical physical response that is generated by the plant to this stress. Whatever we discussed so far will be in a form of a summary for the next slide. So Dipisa, can I please have the next slide? Yes. What is stress? Any deviation from normal condition leading to adverse effect on growth and development of the plant is a stress. Stress inducing factor could be either biological, biotic, or abiotic, that is non-biological, environmental factor. Biotic responses not only have to be quick, but have to be well programmed. Abiotic responses are prolonged responses. The site of attack in biotic is the leaf, whereas responding organ of the plant to any kind of abiotic stress is root. Focusing on abiotic stress, temperature, water, and soil are three identified as abiotic stress inducers. Too low of a temperature, as I said, can lead to freezing stress. Too high of a temperature via osmotic stress experienced by the plant can lead to drought. Too low of a water can also be causing a desiccation, osmotic stress, and lead to drought. Or too high a water can be the cause of flooding stress. With reference to soil, accumulation of high amount of salt will increase the salinity of the soil, and the plants with high degree of salinity in the soil will get exposed to salinity stress. The topic for my discussion for today and the next lecture and the third lecture shall be salinity stress. So we will focus on salinity stress. Terrestrial higher plants come across a high concentrations of salts when they are growing either closer to seashores or they are growing in estuaries and constitute what is known as estuarine ecosystem. I hope you know estuaries are the places where there is a mixing of sea water, marine water, salt water with the fresh water. This mixing, this replacing of both with each other is subjected to the tidal waves. Now, at such locations, which are estuarine regions or close to seashore, where there is a mixing of fresh water with the salt water, during the resting time of tides. Now, what do you mean by resting time of the tides? It's a time between two successive tides. During this resting period, two processes happen. One is evaporation, and the second one is transpiration. Now, both these processes together will remove the pure water from the soil and give it to the environment in the form of vapors. So now soil is subjected to water loss. And as it is, the soil is representative of estuarine region where there is a mixing of marine and fresh water. This will therefore increase the salt concentration in the soil. This serves as a cause of increased salinity in the soil. Evaporation and transpiration, making loss of water to environment in the form of vapors that indirectly concentrate salts, and such saline soils will then be the cause of salinity stress. There are several properties that these saline soils exhibit. For example, they turn to be less alkaline, less than pH 8.5. Due to the salts, they have high degree of conductance, and obviously their osmotic potential value drops down because the solute potential tends to be more. Well, at this point of time, students, I must clarify two terms because when you go back to books, these two terms will be there. One is called a sodosity, and in comparison to that, salinity. Sodosity refers to the presence of high amount of sodium in the soil. But when you talk about the salinity, you are talking about all total salts, which includes sodium chloride, but it also includes calcium, magnesium, sulfate. Obviously, the estuarine regions receiving large amount of marine water through the tides get enriched in sodium chloride. And most of the plants cannot survive in a soil with sodium chloride more than 200 millimolar. In fact, you must have come across the classification of plants into glycophytes and halophytes. Halophytes are the native to saline soil, and these are the plants which are capable of completing their life cycle in that environment of high salinity. On the other hand, glycophytes are not able to resist the high amount of salt to the same degree as the halophytes. High amount of salt adversely affects their growth and the productivity of the plant is less. Now the question here is, The plants which or otherwise are showing healthy growth, why are they not able to grow in the soils with high amount of salt concentration? What is the reason? And related to this, therefore, the second question arises that if there are definite reasons to which the plant is not able to grow in such soils, how is that helophytic plants have adjusted themselves to this stress? What are the ill effects, therefore, of a salinity, which as said, limits the growth of a plant in saline soil? Let us examine them. One, saline soil or salinity in the soil induces osmotic stress. Now, I hope you all understand when I use the word called osmotic stress, water potential, solute potential, inverse relationship between the two, definition of osmosis, hypotonic and hypertonic solutions. Dissolved salts in the water of a soil solution makes it hypertonic. Now, when the environment outside turns hypertonic, the water absorbing capacity of the plant in that soil decreases. Decrease in osmotic potential. And that decrease causes a stress to the plant. And the plant does not have minimum absorption of water. That's how salt interferes. Second, high amount of salinity leads to ionic imbalance. Now, this aspect also indirectly we have learned in the lower class. When we discuss the passive transport and active transport, we talk about Donan equilibrium, carrier concept, etc. The inner cellular environment of the plant tries to maintain an ionic balance. Balance not only between the absorbed ions, but balance also with the ions of the outer environment. Now, if this interdependence is existing, then disturbance of ions in any one phase can be the cause of disturbance in the overall ionic balance. Salinity is contributed by calcium, potassium, sodium, sulfates, magnesium. So this salts interfere with the management of ionic balance that a plant tissue was having in accordance with the outer environment within itself in its different compartments. For example, you know the vacuoles are the sites available in the cell, the largest compartmental size as a reservoir of salts. All this is balanced for the plant. It is put into imbalance by for the plant. Added to this, the membrane proteins we shall talk about which little later, called the transporters, symporters, antiporters, coporters, etc., also have disturbed functioning. The third reason, oxidative stress induced by salinity. You are aware of the functioning of electron transport system for both mitochondria and chloroplast. It's a series of temporary electron carriers with the proton channels working with important enzyme cofactors, leading to synthesis of energy-rich compound and ultimately leading to oxygen trapped in water. High level of salinity tends to interfere with this ETS functioning and becomes a cause of ROS, that is reactive oxygen species, such as singlet oxygen or hydroxyl ion radical or H2O2. And these free radicals then target the healthy cells, and cause the damage, which is irreparable. So this oxidative stress can also be experienced by a plant under high salinity. The fourth reason, adverse effect of salinity on growth. How? As I just now mentioned, plants have a very coordinated method by way of which they not only identify the signal, not only sense the signal, but transfer that signal to transcriptional factors, which then generates the necessary response via signal transduction pathway. And any kind of abiotic stress, the signal transduction pathways tend to increase one hormone level, and that is ABA, abscisic acid. Abscisic acid has a tendency to lead partial closing of stomata. So obviously under this condition, the plant growth is affected because ABA also has a certain inhibitory effect on general growth and morphogenetic processes in plants. Fifth, adversely affecting the photosynthesis. As I said, increased level of ABA tends to partially close the stomata, which facilitates a less amount of carbon dioxide entering the leaf tissue, and that affects the photosynthetic ability of the plant, and in long run, the productivity of the plant also drops down. So high level of salinity causing osmotic stress, causing ionic imbalance, causing oxidative stress, adversely affecting photosynthesis in the growth of the plant is reason why plants are not able to sustain this saline environment. But then as I put a question to you, if it is difficult for one group of plants, how is it possible for another group of plants called halophytes? One, any abiotic stress is a prolonged stress. This provides an enough opportunity for a plant to adapt itself to the adverse situation and respond to it in a favorable way in favor of its own growth in future. What I'm trying to say is that mangroves are the unique species which have over the years adapted themselves to the saline environment. So this adaptive mechanism of survival is what is helping these halophytic plants to complete their life cycle in the saline environment. Secondly, mangrove forests are identified also by the presence of pneumatophores, negatively geotropic breathing roots which rise above the ground surface, and with the lenticels they grab the oxygen of the environment and transfer it to the roots which it requires for respiration, but under marshy, loggy soil, non-availability of oxygen due to anaerobism is thus compensated, and the plants are able to sustain their energy requirement via respiration and thereby the overall growth. Another characteristic which can be considered as a characteristic of prime importance for mangroves to be able to complete their life cycles in saline environment is the vivipary. What is vivipary? Method wherein seed germinates while it is still on the fruit. Unlike other plant species, in mangroves, the seeds are not dispersed to either narrow or a wider distances, which then fall on the soil and germinate. Mangroves are saline plants. High degree of salinity will make less water available, and water being the essential requirement for seed germination will be hampered. But what mangroves have adopted a strategy? They allow the germination of seed, let the propagule be formed, and then that propagule is dropped close to the parent plant in the soil. A propagule will have definitely better mechanism to root itself and survive in the environment of salinity rather than the seed, which will be lost immediately due to non-availability of water. So long-term adaptive strategy for survival, pneumatophores, vivipary mode of growth of seed germination followed by growth is what makes them unique and surviving plants in saline environment. Many of the mangroves can go up to the level of 300 millimolars of sodium chloride and still managing to grow. Uh, Dear students, one thing I would like to stress here before I discuss the slide which is already shared by the PISA. If we happen to go to a mixed population of mangroves for a visit, well, one has to be very careful because the soil is so very soft and marshy that you really have to be very careful. You should be ready to get dirty to go to the mangrove forest, as they say. But when you happen to look at the mangroves, with little bit close examination of mangroves, in terms of morphological appearances or anatomical features, you can divide them into certain categories. I'll tell you what I'm trying to say. Certain mangroves, morphologically, in terms of their habit as well, will give you the characteristics of xerophytic plants. What are xerophytic plant characteristics usually? Reduction in the leaf to spines, or reduction in the leaf number, size of the leaf, then sunken stomatas, multiple epidermis, well-developed trachea elements, etc., etc. So xerophytic characters dominate in some of the halophytes. That is what you'll be able to observe. Another group of halophytes, very luxuriously growing, having a wonderful habit, looking like a natural plant, other plant, non-mangrove plant, but the undersurface of the leaf when you examine will appear dull. And that particular surface will also have a layer of salt that you can experience if you pass your fingers over the lower epidermis. And another group of mangroves which will distinctly catch your attention is that you'll be able to see the long stems with the exposed naked nodes, and terminally, apically, a bunch of young leaves. Lateral branches also a bunch of young leaves and all. All are mangrove species. All are growing in the same habitat of saline environment. But the morphological and anatomical differences at the first instance is primarily due to the fact that mitigation, managing of saline environment is handled by mangroves in more than one way. Managing the salinity by mangroves is achieved by two ways. One, avoidance, and second, tolerance. Avoidance method is a method wherein the plant, mangrove plant, knowing the ill effects of high amount of salt, will reject the salt right at the absorption site of root. If not able to do that, will at least try to get rid of the salt by suitable means before it starts interfering with the metabolism of the plant. On the other hand, the accumulators are those which will show acceptance towards the salt, but then try to accumulate them in structures or the organs which can be easily removed. I am pointing your attention to three type of mangroves classes called excluders, excretors, and accumulators. Excluders are those which exclude taking the salt. They will have to therefore manage with less amount of water. They are okay with it because rather than taking the salt inside and then developing mechanisms to fight with the ill effects of that salt and nullifying it, might as well manage with the less amount of water. That's the reason certain group of halophytes show you xerophytic characteristics, because they are excluders which are happy with less amount of water, because the advantage they derive from this is they do not take any salt. Those group of halophytes I said appear to be quite luxuriously growing, but the undersurface of the leaf has a salt, are the excretors, because on the undersurface of the leaf they develop salt glands, and through these salt glands they will exclude the salt. Because as the water evaporates, the salt will also be evaporated, be returned back to the environment, and the plant will get rid of it. What are the third categories? Where I said that the nodes appear naked and only the bunch of young leaves are the primarily accumulators. They have chosen to accumulate these extra salts by transferring them to the leaves, because as the plant grows bigger, the older leaves are shedded off, and the plant will therefore be able to get rid of unwanted salt. So, well, Deepi sir has already put the next slide for your reference. This is what we have been discussing in the last couple of minutes. What makes the soil saline? I said in estuarine and the seashore conditions, evaporation and transpiration processes tends to lose the water and thereby indirectly concentrate the salts in the soil. What is the effect of soil salinity on the plant? Well, glycophytes are unable to survive in salt concentration, because primarily the high amount of salinity interferes with the metabolism, in the sense that it reduces photosynthesis, it reduces growth, it induces osmotic stress and oxidative stress, and something which is very important at a cellular level for a plant to maintain the ionic balance. That ionic balance is totally disturbed. Mangroves, however, are able to survive in saline environment. Beautiful group of plants for the study, because they have developed mechanisms by way of which they can survive in this environment. Special mention is to be made about their adaptive mechanisms in terms of tolerance and avoidance. Special mention is also to be made about the pneumatophore structures that they give out and the vivipary mode of germination. So mitigation of mangroves is, in mangroves, the mitigation of salinity is done either by salt avoidance and salt tolerance. And these salt tolerance are the accumulator species, and the excluders and exciters are exhibiting the means of a salt avoidance. In today's lecture, we will be focusing now on salt tolerance. We'll be focusing now on salt tolerance. Well, how do I define salt tolerance? Salt tolerance can be defined as ability of a plant to tolerate the salt without letting it affect the growth, without any adverse effect on the growth. And when I talk about the salt here, I'm basically talking about sodium chloride, and the levels that I'm talking about is close to 200 millimolar to 300 millimolar. Well, if I talk about the estuarine region, when I talk about the seawater, yes, I'm also obviously talking about sodium chloride. Literature defines sodium as highly toxic, and therefore sodium accumulation could be toxic for the plant. Why? One, sodium prevents adequate moisture uptake by the plant root, and thus, two, it leads to root desiccation. One and two both leads to water stress experienced by the plant, and one, two, three together therefore subjects the plant to physiological drought. Try and understand this combination of words. Physiological drought. Drought is a condition of desiccation where there is a scarcity of water. By calling this drought here as a physiological drought, it means that water is available in the surrounding environment, but not available for absorption due to reasons of high salt. This drought, therefore, will be a physiological drought. And as a combined or cumulative effect of all this reduction in the plant growth, this justifies the sodium toxicity. But in that case, if a salt tolerant plant species is going to be tolerant to the sodium chloride in the initial phase of absorption by roots and not rejecting the way the extruders are rejecting it or excretors are excreting out, then what is the immediate response they show so that they prevent their tissue from sodium injury? One of the effective way I described to you, they transfer it straight to the leaves, and those leaves when lost, that excess salt is also lost. They can't afford to do this all the time because we are forgetting that leaves are the main photosynthetic site generating food for the plant, and its survival is dependent on that. So many a times they compartmentalize them to portions of the stem which do not come in direct contact with the metabolic site of leaves. Another effective mechanism that these plants show is direct transfer of sodium to the vacuoles. Vacuoles, as I said, are the large storage compartments of the plant cell, and transporting the sodium is temporarily removing the interference of it with the normal metabolism and the cellular reactions. What is also very important here in managing the sodium toxicity is the role of transporters. You know, there are certain membrane proteins which help in transport of ions across the membrane. The membrane which is separating the soil solution or the cellular environment of the cell, but this also includes the intracellular membranes like tonoplast of the vacuole against the cell membrane. We'll talk about this. Can I have the next slide, please, Dipisa? Yes. I said today we'll focus on salt tolerance. And here is the list of different biochemical and physiological methods exhibited by halophytes towards a salt tolerance. Let us take the first, ion homeostasis. Now, what do you understand by ion homeostasis? It is defined as the mechanism of plant to maintain internal steady state of ions. Ability of a plant, mechanism of a plant to maintain the steady internal state of ions. I hope all of you know that in plants, most of the membranes are energized with the gradient of protons. Gradient is concentration, concentrated with protons. Can anyone tell me? Can any one of you tell me as to why do I need a higher proton gradient on the membrane surface? I need a higher proton gradient on the membrane surface. Primarily is for the functioning of enzymes like ATPases. You know, ATPases are the membrane-bound allosteric conjugate enzymes, the active sites of which have to be blocked by protons so that allosteric sites open up, which are then offered to phosphates to complete the role of synthesizing the energy currency called ATP. So high amount of proton is rendered at the membrane surface. Secondly, into the membrane, there are proteins which work differently in terms of entry or exit, efflux or influx of ions. For example, uniporters, channels which function along the gradient, symporters, transporting ions in the same direction, antiporters, transferring ions in different directions, and so on. If a plant needs to maintain its ionic balance undisturbed, that means it is requirement of a cell to also maintain its electro neutrality so that the metabolic reactions taking place within the cell can continue to fix at non-fluctuating pH. I said a sodium detoxification effective method is also to transfer them to vacuoles. A soon stage would come when the vacuole would have a requirement of water, and the requirement of water by the vacuole for the osmotic adjustments has to be compensated by the cellular environment, and cellular environment will have to depend on outer soil environment, which in this case is highly saline. Now take the combination of transporters on the membrane and the high proton gradient on the membrane. Sodium proton antiporter, a protein which will throw the sodium out and in return will accept the proton in. They partially help in removing sodium toxicity and maintaining the electroneutrality of the cellular environment. You will say as neutron proton as sodium is going, the protons are also entering in, but those protons are utilized by the membrane bound enzymes like ATPases. So in a way, the disturbing component of sodium is taken care of, and the neutrality or constant ionic status is maintained for the cell. In nutshell, special membrane proteins working in both antiporter way or symporter way, membrane channels which allow the flow of ions along the concentration gradient, and the higher proton rendering on the surface of the plant cells come together and help the plant in overcoming the ionic imbalance, or in other words, help the plant in setting ion homeostasis. The second method of salt tolerance is the regulation provided by osmolyte accumulation. What are osmolytes? Osmolytes are a group of chemically diverse organic compounds that are uncharged, polar, and soluble in nature. They do not interfere with the cellular metabolism even if they are present at higher concentration. What are they meant for? They are meant for osmotic balance. Now, what did I say? I said that compatible solutes are a class of compounds which are polar and soluble. They have high affinity for water. They are uncharged neutral, so chemically don't interfere with the cellular metabolism. And they help in osmotic balance. The logic is simple. These osmolytes or compatible osmolytes or compatible solutes as they are named as will increase the solute potential of the plant cell. Correspondingly, water potential will be less. This will allow the influx of water along the gradient from outside to inside, and the plant will be able to have its osmotic adjustments, and to an extent will also be able to dilute the effect of salt. Are there any compatible solutes chemically identified as osmolytes and performing any other role? Yes. One such compatible solute or the osmolyte is a proline. Studies indicate that during a salinity stress or otherwise the amino acid makeup of the plant gets poor. Majority of the free amino acid, the concentration during salinity decreases. But one amino acid proline shows concentration rise during salinity. Now, the reason to this effect is that one, it serves as an osmolyte. So obviously it helps in the osmotic adjustment that the plant is in the deadly need of due to high amount of salinity. But under the stress, when it could be under severity where the plant existence, survival is questionable, at that time, these prolines can serve as reservoir of organic nitrogen. So proline as an osmolyte, and during salinity, proline as an organic nitrogen reserve is how we remember its role. The second compatible solute is glycine betaine. It is an amphoteric quaternary ammonium compound. What role it has to play? Well, I have identified it as osmolyte. So it is going to help the plant in osmotic balance by serving as a solute. But it also plays a very important role in protecting the photosynthetic apparatus of the plant during salinity. Again, it goes back to the same argument that in future, the intensifying of the stress may come to a stage where the plant will have to have a question of survival. At that time, the intake photosynthetic apparatus can redeem the plant for generating food. Well, glycine betaine is also very important during salinity stress because it can act as compound leading to reduction of ROS. I said reactive oxygen species that are generated during oxidative stress induced in salinity. Combating them can be easy by glycine betaine. The third important osmolyte after proline and glycine betaine is polyols. As the name suggests, these are the multiple hydroxyl functional group containing compounds, polyols. So multiple hydroxyl functional group containing compounds. They are of two types. One type is called cyclic polyols. You heard the names penitol, myoinositol, and the other ones are acyclic polyols like mannitol, sorbitol, etc. What function do they perform? Well, I'm talking about them as osmolytes. So number one, osmotic balance. Any other function attributed to it during a salinity stress? Yes. They act as scavengers of reactive oxygen species. Any other function? Yes. They help in stabilizing the enzymes, and very important, they also help in maintaining membrane structure. The fourth compatible solute is a sugar carbohydrate. Sugars such as glucose, fructose, starch, they occur under salt stress. What role? Again, an osmolyte, except for starch, again an osmolyte. So thus they help in osmoprotection. But apart from that, they also help in scavenging of ROS. And very important, these are carbon derivatives which serve as carbon storage reserves, the way the proline serves as organic nitrogen reserve. So methods of salt tolerance, ion homeostasis, taking advantage of high proton gradient on the cell, taking advantage of different transporter proteins available on the membrane, taking advantage of intercompartmental distribution of sodium. Plant cell tries to regain its ionic balance by means of ionic homeostasis. Osmolytes, if accumulate in the cellular environment, will tend to increase the solute potential, by way of which they will be able to attract more water from outside. Thus, they play a role in osmotic balance. But each one has, apart from osmotic balance, a specific other role as well. For example, proline is also organic nitrogen reservoir. Glycine betaine protects the photosynthetic apparatus and effective against ROS. Polyols, they stabilize the enzymes, membrane structure they maintain, and effective against ROS. Sugar carbohydrates are again the carbon storage reservoir, and they are effective against ROS. The third method is the method imposed or method based on regulation by antioxidant enzymes. It's quite simple for you to understand after the discussion that we have been having for the last 1 hour. As I said, the ROS species that are generated due to malfunctioning or no function or disturbed, interrupted functioning of ETS. These have to be quenched somehow, otherwise the chain reactions that they lead to can damage the healthy cells. So what comes into picture here is a galaxy of antioxidant enzymes and important antioxidant enzymes. The names that you can remember combating the oxidative stress during salinity is superoxide dismutase, SOD, then CAT, which is catalase, then GPX, which is glutathione peroxidase, and APX, which is ascorbate peroxidase. Since they help the plant in overcoming the oxidative stress, these all enzymes, rather than just calling them as an antioxidant enzymes, in this case will be labeled as regulatory enzymes. Role of polyamines. What are polyamines? Polyamines are a small, low molecular weight, widely distributed in plant system, ubiquitous, polycationic, that means having multiple positive charges, molecules, which are, as I said, widely found in plant kingdom. If they're widely found in the plant kingdom, do they have any specific role assigned to them? Yes. They have roles assigned to them. They participate in more than one functioning of seed germination, dormancy breaking in tubers, embryo development, differentiation during the growth, morphogenesis, development of flowers and the fruits. But these polyamines are important to discuss in salt tolerance because they also serve as the indicators of severity of salinity stress. See, in a way, it also can be classified as a compatible solute or osmolyte. But higher amounts of polyamines is an indicative of the severity of the stress. So that's why polyamines and their profile during salinity is important to study because it will be generated only towards the severity. So indirectly, one can correlate the severity of the salinity with higher or lower concentration of polyamines. Diamine putrescine, that is PUT, put, then triamine spermidine, that is SPD, and tetraamine spermine, that is SPM. These are the types of polyamines. Of course, the studies are with the in vitro salinity stress induced in plants like rice, tobacco, and Arabidopsis. But with reference to mangroves, the reason for their sudden increase during the acute salinity stress is yet to be understood, but they do serve as a candidate which can describe to you the severity of the stress. Well, coming to the last role of hormones, I think I did mention to you that the plants have very ordered mechanism of identifying the signal and creating necessary response through signal transduction pathway. And during this signal transduction pathway, increase in the abscisic acid is observed in most of the abiotic stresses, whether it is a water stress, temperature stress, or a light stress. But what is very interesting here is to note that increased level of abscisic acid, how does it help the plant in a salt tolerance? Well, increased elicited ABA level tends to increase the activation of certain genes. And these genes, under modulation by abscisic acid, will lead to active production of certain osmolytes like proline, sugars, and will also initiate the higher accumulation of potassium and calcium in the cell. So this helps the plant cell to have the increased osmolyte level due to the genes activated by high amount of ABA. That's the role of ABA plays. And it also helps the plant to accumulate large amount of potassium and calcium. How effectively this potassium and calcium can be used by the plant to counteract the ill effect of sodium, that will definitely be get highlighted in the next lecture when we meet. However, apart from abscisic acid, it is also the salicylic acid, and in certain cases, like plants like rice, barley, and Arabidopsis, again, the studies done by in vitro salinity exposure, the brassicosteroids, BR, are also believed to be the hormones that increase during salt tolerance. So a quick review of biochemical physiological methods of salt tolerance: ion homeostasis, regulation of osmolyte accumulations. I given you 4 names of those osmolytes, if you remember: proline, glycine, betaine, polyols, and sugar carbohydrates. Regulation by antioxidant enzymes. We spoke about SODs, talk about CAT, APX, and GPX. Polyamines under severe salinity stress is exhibited by them, and types of polyamines that we are talking about is PUT, SPD, and SPM. And hormones like abscisic acid, salicylic acid, and brassinosteroids are also helpful to the plant in physiological and biochemical methods of salt tolerance. Can I have the last slide, please? Dipisa? Yeah, thank you, Dipisa. In the coming lecture, uh, we will focus on what is known as a SOS pathway, a beautiful pathway proposed by a physiologist explaining how salt tolerance can be mitigated by ion homeostasis. And after completing that discussion, we will talk about different methods of salt avoidance after doing today's salt tolerance, and bacteria which can survive under highly saline environment. Prokaryotic organisms without any distribution of function or distribution of labor to the cellular compartments, and yet sustaining the high amount of salinity. How? That is the beauty of the topic which we will deal with. So with this, I end my lecture today. Thank you, everybody. If you have any questions on the topics, please note them. In the last session, I will answer all your questions. Reference books, Buchanan, E-Pathshala notes, and Levitt. That is what I have referred to. I will also give you the printout of these slides with the important points incorporated that will serve as ready reckoner to you in time to come. We will also have the notes for your reference in the final exam. So thank you very much, all of you. We begin with the next lecture, uh, which is by Aishwarya Madam, paper 2. Dipisa? Dipisa?
Hello.
Yeah, Dipisa, better thank you very much.
Yeah.
But I have a small, Dipi, could you?
Okay. Hello students, welcome. Welcome to this online platform of this virtual teaching. Okay. So in semester 3, I'm going to take your paper 2. Okay, that is entire paper 2 of molecular biology. So in molecular biology, we study about biomolecules. Okay. These biomolecules are macromolecules. There are many micromolecules in an organism, but here we are interested in nucleic acids and proteins. Okay, so DNA, RNA, and proteins. So in paper 2, we have 4 units. First is DNA replication, second is transcription, third is RNA processing, and fourth is translation. Okay. Now, today I'm going to give you an overview of the paper, and I want to discuss some basic points, or let's say some revision before we start with the actual topic. Okay. Now, we can put this syllabus, entire syllabus, in one diagram. Okay, you must have heard central dogma. Okay, so what is central dogma? In central dogma, first is DNA, that is a genetic material. If DNA gets converted into DNA, it's called replication. Okay, that's your first unit, that is DNA replication. Then DNA gets converted into RNA, that is transcription. Then that RNA gets translated to form protein. The entire process is called as central dogma. Okay, you can see DNA is double-stranded molecule, RNA is only single-stranded, and then after translation, it forms a three-dimensional protein. Okay, first obviously it forms polypeptide chain, and then it forms a three-dimensional protein. This entire process can also be called gene expression. Okay, when the gene, that is a segment of DNA, gets transcribed to RNA and then it translated into protein, the entire process, when we say that gene is expressed. Okay. Now, if you look closely in this diagram, your unit 1 is there. Okay, DNA to DNA is replication. Then unit 2 is transcription, that is from DNA to RNA. and unit 4 is there, that is from RNA to protein. Okay. Now unit 3 is not there, that is RNA processing. We'll see later. Okay. Now in molecular biology, you will encounter with the difference between prokaryotic and eukaryotic cells mainly. Okay, because the molecular molecules which are present in plants and animals are more or less same because they both are eukaryotes. So we'll encounter difference between prokaryotes and eukaryotes. Okay, so here we are interested in first main difference that gives a name to these organisms, that is prokaryotes and eukaryotes. Prokaryotes means the primitive nucleus. Okay. So in this left side diagram, you can see the prokaryotic cell. Okay. In the prokaryotic cell, you can see the nucleoid. Okay. Nucleoid means nucleus-like structure. Oid means like. So it is nucleus-like structure. It is not the true nucleus. Okay. So when you see it's a true nucleus, because this nucleoid doesn't have any nuclear membrane, nucleoplasm, nucleolus, and genetic material. Only genetic material is there. Rest three parts of the true nucleus are absent. So this true nucleus is not there. Okay. Only the genetic material is present in that region that we call as nucleoid. Okay. While in right side, you can see a eukaryotic cell. You can see a proper nuclear membrane. Then inside it is nucleoplasm, that blue color region. Then you can see a nucleolus, red one, and there is a genetic material inside. Okay, so this is a true nucleus. Okay, now remembering this difference, we can put the earlier diagram of central dogma like this. Okay, so here you can see there is a boundary of nucleus and cytoplasm. Now you can see that diagram has been split. The DNA to DNA, that is replication and transcription, takes place in the nucleus. Okay, so RNA is formed in the nucleus and then translation takes place in the cytoplasm on ribosomes. Okay, now in prokaryotes, because nucleus is not there, okay, because nucleus is not there, the RNA which is formed comes directly in the cytoplasm. So simultaneously translation starts. So in prokaryotes, it's more like simultaneous transcription and translation. Okay, so there is no time in between these two processes. Okay, but in eukaryotes, because nucleus is there is a time to process the molecule of RNA which is formed in the nucleus. Okay, so here is your third unit, that is RNA processing. Okay, so the first three units, that is replication, three processes, transcription and RNA processing takes place in the nucleus. Okay, after transcription, the RNA molecule which is formed in eukaryotes is processed, that is called as RNA processing. Okay, there are many processing steps that we'll see later in that particular unit, and then that processed RNA molecule then comes in cytoplasm. Okay, and then it gets translated on ribosomes. So RNA processing takes place in proper RNA processing takes place only in eukaryotes because the nucleus is present. There is a time or opportunity to process that RNA molecule in only in eukaryotes. Okay, so this is an overview of your syllabus. Okay, now let's revise some basic points. First is the composition of DNA. Okay, now nucleic acid, that is DNA and RNA both, are made up of three components. First is pentose sugar, second nitrogen bases, and third is phosphate group. Okay, so pentose sugar, there are two types, ribose and deoxyribose. The ribose is in RNA and deoxyribose is in DNA. Okay, you can see the difference between these two sugars here. There are 5 carbons, 4 are in the ring. You can see first, second, third, and fourth, and fifth is outside the ring. Okay, in both cases. Now in ribose, we can see the second carbon, there is OH group. Okay, hydroxyl group at the second carbon of ribose sugar, while in deoxyribose, there is only hydrogen. Okay, so no oxygen. So one oxygen less, hence it is deoxyribose in DNA. Okay. Now you will remember this 1, 2, 3, 4, and 5, the arrangement of the ring. Okay. How the molecule, these functional groups are attached. Okay. Remember this. The next is nitrogen bases. There are two types of nitrogen bases, purines and pyrimidines. Purines are two, adenine and guanine. These are double ring structures. Okay, and pyrimidines are three, cytosine, thymine, and uracil. These are single ring structures. Thymine is in DNA and uracil is in RNA. And the third component is phosphate group. You can see that a lot of negative charge in the phosphoric acid, which is a phosphate group. That's why your nucleic acids, that is DNA and RNA, both are negatively charged. Okay. Now, these three are components of DNA. These three will join together to form a monomer because DNA is a polymer. So every polymer is made up of a monomer. So in DNA or a nucleic acid, monomer is a nucleotide. So here you can see a DNA nucleotide and RNA nucleotide. Now focus on the DNA nucleotide. You can see the sugar molecule. You can identify first, second, third, fourth, and fifth carbon, which is outside the ring. Okay. You can see to the first carbon, base is attached. That is nitrogen base. Here, in case in this diagram, it is adenine. Then at the fifth carbon, which is outside the ring, there is a phosphate group attached. Okay. So first and fifth carbon is engaged. Okay. Now in RNA nucleotide, the arrangement is same, only the sugar is different, that is of ribose sugar in RNA, and the base is uracil, which is only in RNA. Now, you can see in the nucleotide, first and fifth carbon is attached, okay, to their compounds. Then these monomers form polymer. It forms a DNA strand. So in right hand side, you can see three nucleotides, okay. Here, what has happened? The third carbon of first nucleotide is linked to the fifth carbon of the next nucleotide with a phosphodiester bond. Okay, so you can see that phosphodiester bond, there are two ester bonds with the phosphate in it. It's called as phosphodiester, RCOOR. Okay, there are two RCOR bonds. Hence, it is a phosphodiester bond. Now you can see in this diagram, there is a 3' end, that is 3' carbon, and here it is 5th carbon, okay, to every nucleotide. Okay, you can see a third carbon, 5th carbon, third carbon, 5th carbon. Hence, a single DNA strand has two ends. We call it as 5' end and 3' end. Okay, so this is how the phosphodiester bond is formed. So this is only about the phosphodiester bond, but the actual DNA looks like this. Okay, so this is a molecular model first, that is three-dimensional diagram. Second is stylized, and third is chemical structure. So in the first diagram, that is three-dimensional, you can see how the molecules are arranged in the three-dimensional structure. Look at the backbone and look at the central part in which there are base pairs present. Okay. In the second diagram, you can see the backbone is now in the ribbon shape. Okay. And in this diagram, minor and major grooves are distinctly visible. Okay, this is a characteristic feature of double helix of DNA, that is minor and major groove alternate with each other. In this diagram, if you imagine the hypothetical axis passing through the DNA, then the width is 1 nanometer. So the width of entire DNA is 2 nanometers. Okay, remember this. The distance between two nucleotide bases, look at the top of the diagram, it is 0.34 nanometers. And the one complete turn of DNA, if we assume here, in this diagram 10 base pairs, then the total distance becomes into 10, that is 3.4 nanometers. Okay, so one complete turn takes about 3.4 nanometers. Okay, and now you can see in the center all the nitrogen bases are present. In the third diagram, which shows the chemical structure, you can see the two strands of the DNA. Okay, that is the backbone is made up of sugar phosphate molecules. Okay, alternate sugar phosphate that forms the backbone of the DNA, and center you can see the nitrogen bases. Okay, now you know that backbone is made up of phosphodiester bond, which I already saw. Center, the nitrogen bases are linked by hydrogen bonds. Okay, between adenine and thymine there are two hydrogen bonds, and between cytosine and guanine there are three hydrogen bonds. Okay, the two strands of the DNA, look at the polarity. Okay, one strand is in 5 prime to 3 prime direction, while the other you can see it is inverted. So it is 5 prime to 3 prime. Okay, from down, from up it is 3 prime to 5 prime. Okay, you can see that inverted direction. So two strands of the DNA are anti-parallel in nature. Okay, between a sugar and a nitrogen base, that is the first carbon, when the bond is formed, that is glycosidic bond. Okay, between a sugar and a nitrogen base at the first carbon of sugar, there is glycosidic bond. Between a phosphate and the sugar is phosphodiester bond, and between two nitrogen bases there is hydrogen bond. Okay, as you know that hydrogen bond is a weak bond, it is easy to separate two DNA strands. Okay, because two DNA strands are joined only by hydrogen bonds. Hydrogen bonds are weak bonds, so change in temperature, pH, will separate two DNA strands. Okay, but it is difficult to cut DNA strand because you can see the backbone is made up of phosphodiester bond, which is a covalent bond. So for cutting DNA strand, you need enzymes. You must have heard of nucleases. There are different types of nucleases which cut the nucleic acid. So we need enzymes to cut the DNA strand. But while to separate two DNA strands, it is easy because only hydrogen bonds are present. Okay, so this is about the structure of DNA. So these are some characteristic features. So DNA molecule has two polynucleotide chains bound around each other to form a double helix. Diameter is 2 nanometers. They are anti-parallel in nature. The two strands are made up of sugar phosphate backbone. Then there is complementary base pairing between adenine and thymine as well as guanine and cytosine. Uracil is present only in RNA, and uracil pairs with adenine. Okay, there are two bonds between A and T and three in between G and C. Then remember the distance. It is 3.4 nanometers for 10 base pairs. Okay, we are considering that 4 turn has 10 base pairs of the of nucleic acids. Okay, of nucleotide base pairs. Okay, the complete turn is 360 degrees. So each base pair is twisted 36 degrees. That's why here you can see the twisting of the DNA in first and second diagram. The twisting is because of the 36 degrees of the base pair. Okay, 36 degrees. So 36 degrees, the base pair gets turned. That's why the DNA forms a double helical structure. Okay, and the last is DNA double helix has major as well as minor group. So all these are characteristic features of DNA. Okay, remember all these. Then what is the function of DNA? Okay, this all was about structure. So what is the function of DNA? DNA stores an information which codes for a sequence of amino acids to form protein, right? During transcription translation. So what DNA possesses? It possesses the information. So the entire information is function. That means you have only genes in the DNA? No. Okay. Here is the pie chart which shows the human genome. Okay. In human genome, you can see There are 30% genes, 45% transposons. Now you know transposons, we already done, okay, the jumping genes. And the 25% is miscellaneous. Okay, that is the other type of DNA. So you can see only 30% of the entire genome of our genome is genes. Okay, now in that 30% also, you can see only 1.5% is exons. Now you must have heard exons and introns. Okay, in gene, when the, as because we are eukaryotic organisms, our pre-mRNA, that is mRNA after transcription, will have exons and introns both. Now, introns are intervening sequences and exons are expressed sequences. Okay, so introns are removed and exons are joined together. So exons are the only sequences which actually forms the protein. Okay, which actually gets translated into a protein. Okay, so you can see only 1.5% is exons. So from the entire genome, only about 1.5% is exons. Rest all is, you can say it is non-coding. Okay. So in genes, you can see 28.5, the rest is introns and non-coding segments. Okay. So introns you already know. The rest non-coding segments will be all the regulatory DNA. Okay. When you say gene has regulatory sequences and coding sequences. Okay, so your introns, they get transcribed, they are a part of your mRNA, but other regulatory sequences, they are not part of mRNA. Okay, they do not get transcribed. Okay, so what are these regulatory sequences? The promoters, enhancers, silencers, which activate the gene expression or which inactivate or repress the gene expression. All these sequences, they are non-coding segments. Okay, different proteins bind to these segments, these sequences, and they regulate the gene expression. They decide how to how these gene gets expressed, how particular gene gets expressed. Okay, so all these are your non-coding segments in the gene. Okay, so you can see the difference. If, for example, suppose 100 base pair is the gene, 400 base pair could be regulatory sequence. Okay, more sequences are for regulatory sequence than the actual gene, remember. Okay, that's why this much of difference. So the entire genome, only very few are exons. Okay, transposons, you already know, lines and sign sequences, long interspersed elements, short interspersed elements. Okay, and miscellaneous is the other DNA, that is your simple repeats, segment duplications, and some DNA which forms RNA. Okay, in gene expression, we only talk about mRNA, but you know there are other types of RNAs also, like transfer RNA, ribosomal RNA. There are other types of RNAs also. Okay, so that information is also with DNA. So that all becomes the miscellaneous, because these genes are very, DNA sequences are difficult to locate. Okay, that's why it is in miscellaneous. So this was to give you an overview of a human genome. Then next is DNA to chromosome. Okay, you know that we have seen the structure of DNA, but when in during cell division, DNA is in the form of chromosome. Okay, so how DNA forms a chromosome? Okay, there is a lot of packaging, because you can see the length of the human DNA is very long, and as compared to the human cell nucleus, this is just a comparison. Okay, so for that also, there is some level of packaging is required to accommodate that huge DNA into the small nucleus. Okay, chromosomes are only present, they take the shape of chromosome only during cell division, but still during interphase also, DNA is under some level of packaging. Okay, so here first you can see at the left hand side, DNA double helix, that is 2 nanometer diameter. Okay, you know now it is 2 nanometers. Then first, it gets bound around histones. You must have heard histone proteins. Okay, these are the protein molecules which help in the DNA packaging. Okay, so There are many histone molecules, and four histone molecules forms a core on which the DNA gets bound. You can see in the next diagram there are four histone molecules. Okay, four this side and four out. So to that side, this is a three-dimensional diagram. So you can see the eight. So it is an octamer. Okay, so on this octamer, DNA is getting bound. Okay, here at this level of packaging, this is the second level of packaging you can say, which is a nucleosome, and it is 10 nanometer diameter. Okay, so what is nucleosome? DNA around bound around a octamer of histone. It's a nucleosome. Okay, then in between nucleosome you can see there is a linker DNA. So now this on linker DNA there is one more histone gets attached and it forms a 10 nanometer diameter structure. Okay, this looks like a beads on string model. Okay, so beads are your histone proteins, that is octamer, and string is DNA. Okay, so it looks like a beads on string structure, so we call it like that. Then you can see next, they get bound around each other to form a spiral. Okay, this supercoil is also called a solenoid structure, which is a next level of packaging which forms a 30 nanometer diameter. Okay, now from 30 nanometers, it directly goes to 700 nanometers. Okay, there must be some other levels of packaging, but that is not very distinct. Okay, but remember, when it jumps from 30 nanometer to 700 nanometers, there are other proteins which help. Okay, histones are not the only proteins which help in DNA packaging. There are non-histone proteins also. Actually, here in between this 30 nanometer to 700 nanometer, somewhere there is a non-histone protein core on which the DNA gets wound, or the DNA gets arranged so that it suddenly becomes so complicated. Okay, so there are some other non-histones like cohesins, condensins. There are some proteins which are help in DNA packaging. Okay, now you can see this at the top right, the most right. It is a metaphase chromosome. Okay, that metaphase chromosome, one arm is 700 nanometers, so the entire chromosome is 1400 nanometers. Okay, so this is how the DNA gets converted into a chromosome during cell division. Okay, only at the cell division chromosome is visible. Then afterwards, it usually is in the chromatin form. During interphase, the DNA remains in the genetic material remains in the chromatin form. Okay, because it has to be open for replication and transcription. Okay. Now, the structure of chromosome. You can see the left hand side, the structure of chromosome. Okay, but you must have not seen this type of chromosome. The right hand side, you can see the actual chromosome, which we see the usual diagram of chromosome. Okay, because you know that chromosome is visible only during cell division, and if you imagine a cell cycle, okay, cell cycle, you have studied cell cycle, right? So in cell cycle, first comes G1, then comes S, then comes G2, and then comes M. That is your division phase. So before coming to division, DNA gets replicated. Okay. So that's why you get a sister chromatids in this structure. The actual chromosome is the left hand side, but we will never able to see that because it gets replicated before in the cell cycle. Okay. So in a chromosome, you can see a short arm, that is a P arm, and a long arm, that is Q arm. And these two has a constriction in the center called as centromere. Okay. Now, meros means part in Greek. Okay. So centro refers to central. So it is a central part of the chromosome. Okay. And in the left side diagram, you can see telomeres. Okay. Telos now means end and mere means part. So it is an end part. That's how the chromosome is divided into parts. So central part is the centromere, end part is a telomere. Okay. So both ends of the chromosome has telomeres. Okay, remember these telomeres that will come in the later lecture. Okay, these telomeres are important. Now you can see there are two arms, obviously, short arm and long arm. And in the right hand side diagram of chromosome, blue diagram, you can see the kinetochore. Okay, you must have heard this word kinetochore. Okay, this kinetochore is the complex of protein which is formed during cell division, because in cell division, during cell division, the spindle fibers should attach to the centromere. Okay, so there has to be something which provides a site of attachment to spindle fibers so that it can join to centromere. So that is kinetochore. Okay, this complex forms on the centromere and then spindle fibers attaches to it and then cell division takes place. Okay, they get pulled apart on each pole. Okay, so this is the structure of chromosomes. So depending on the structure, there are types of chromosomes. The first is metacentric, that is when the centromere is the center and two arms are almost of equal length. Okay, then submetacentric, the centromere is slightly away from the center, so one arm is bigger than the other. The next is acrocentric, means centromere is towards the end, so one arm is much shorter than the other. And last is telocentric, means the centromere is at the end. So telos means end, so centromere is at the end, the tip of the chromosome. Okay, so that chromosome has only one arm. Okay, so these are the types of chromosome based on the position of the centromere. Okay, so this entire was some basic points which I want to convey before we start. Okay, now